Cellular receptors are specialized proteins located on the cell surface or within cells that play a critical role in signal transduction. They detect and respond to extracellular signals, such as hormones, neurotransmitters, and environmental stimuli, by initiating specific cellular responses. This study provides a detailed overview of cellular receptors, their subtypes, and a focused examination of G-protein-coupled receptors, GPCRs, including their structure, function, and mechanism of action. 1. What are cellular receptors? Cellular receptors are proteins that bind to specific signaling molecules, ligands, and trigger a response within the cell. They act as molecular switches, converting extracellular signals into intracellular actions. Receptors are essential for maintaining cellular communication, homeostasis, and coordination of physiological processes. 2. Types of cellular receptors. Cellular receptors are broadly classified into four main types based on their structure and mechanism of action. A. Ligand-gated ion channels, ionotropic receptors. B. G-protein-coupled receptors, GPCRs. C. Enzyme-linked receptors. D. Nuclear receptors. A. Ligand-gated ion channels, ionotropic receptors. These receptors are ion channels that open or close in response to ligand binding. They mediate rapid synaptic transmission in the nervous system. For example, nicotinic acetylcholine receptor, GABA receptor.
B. G-protein-coupled receptors, GPCRs. GPCRs are the largest and most diverse family of receptors.
They activate intracellular signaling pathways via G proteins. Examples include adrenergic receptors, opioid receptors. C. Enzyme-linked receptors. These receptors have intrinsic enzymatic activity or are associated with enzymes. They often initiate signaling cascades involving phosphorylation. Examples: receptor tyrosine kinases, for example insulin receptor, guanyl cyclase receptors. D. Nuclear receptors. These receptors are located inside the cell and regulate gene transcription in response to ligand binding. Examples: steroid hormone receptors, say for example estrogen receptor, thyroid hormone receptor. 3. G-protein-coupled receptors, GPCRs. GPCRs are the largest family of cellular receptors, with over 800 members in humans. They are involved in a wide range of physiological processes, including sensory perception, immune response, and neurotransmission. Structure of GPCRs. GPCRs are characterized by a 7-transmembrane, 7-TM, alpha helical structure. They have an extracellular N-terminus, an intracellular C-terminus, and 3 extracellular and intracellular loops. The ligand binding site is located within the transmembrane helices or extracellular loops. Classification of GPCRs. GPCRs are classified into 6 families: AF, based on sequence homology and functional properties. Class A, rhodopsin-like. It is the largest family and includes adrenergic, dopamine, and opioid receptors. Class B, secretin-like. This one includes receptors for peptide hormones like secretin and glucagon. Class C, glutamate-like. It includes metabotropic glutamate receptors and GABA receptors. Class D, fungal-mating pheromone receptors, found in fungi. Class E, CAMP receptors, found in slime molds. Class F, frizzled/smoothened, includes receptors involved in WNT signaling. Mechanism of GPCR signaling. GPCRs transmit signals by activating heterotrimeric G proteins, which consist of 3 subunits, alpha, beta, and gamma. The mechanism involves the following steps. It has 4 major steps and actually starts with ligand binding followed by G protein activation leading to downstream signaling and then signal termination. A. Ligand binding. A signaling molecule, ligand, binds to the extracellular domain of the GPCR, causing a conformational change.
B. G protein activation. The conformational change in the GPCR allows it to interact with and activate a G protein.
GDP bound to the G-alpha subunit is exchanged for GTP, causing the G-alpha subunit to dissociate from the G-beta-gamma complex. C. Downstream signaling. The G-alpha and G-beta-gamma subunits interact with effector proteins to initiate intracellular signaling pathways. OG-alpha-S activates adenol cyclase, increasing CAMP levels. OG-alpha-I inhibits adenol cyclase, decreasing CAMP levels. OG-alpha-Q activates phospholipase C, PLC, leading to the production of IP3 and DAG. OG-beta-gamma can activate ion channels or other signaling molecules. D. Signal termination. The G-alpha subunit hydrolyzes GTP to GDP, allowing it to reassociate with the G-beta-gamma complex and return to an inactive state. GPCR kinases, Brooks, phosphorylate the activated GPCR, promoting the binding of arrestin, which desensitizes the receptor and prevents further G protein activation. 5. Physiological roles of GPCRs. GPCRs are involved in a wide range of physiological processes, including sensory perception. Rhodopsin in the retina detects light. Neurotransmission. Dopamine and serotonin receptors regulate mood and behavior. Immune response. Chemokine receptors mediate leukocyte migration. Metabolism. Glucagon and insulin receptors regulate blood glucose levels. 6. Clinical significance of GPCRs. GPCRs are major drug targets due to their involvement in numerous diseases, cardiovascular diseases. Beta blockers target adrenergic receptors to treat hypertension. Mental health disorders. Antipsychotics and antidepressants target dopamine and serotonin receptors. Cancer. GPCRs are implicated in tumor growth and metastasis. 7. Research and therapeutic applications. Drug development. Over 30% of FDA-approved drugs target GPCRs. Structural biology. Advances in cryo-EM and X-ray crystallography have provided insights into GPCR structure and function. Biased signaling. Researchers are developing drugs that selectively activate specific signaling pathways downstream of GPCRs. Cellular receptors, particularly GPCRs, are fundamental to cellular communication and signal transduction. Their diverse roles in physiology and pathology make them critical targets for drug development and biomedical research. Understanding the structure, function, and mechanisms of GPCRs provides valuable insights into cellular signaling and opens new avenues for therapeutic intervention. Whether you're a student, researcher, or clinician, the study of cellular receptors offers endless opportunities for discovery and innovation.